Good morning. I want to just make a couple of announcements. The first announcement is that labs and discussion sections begin this week. Make sure that you've read the lab so that you can collect the material that was requested for you to collect, and also answer the question which I put up on the board for you in regard to the material that you're collecting for this week's lab. Also, I want to remind you that my office hours begin this week, and contrary to the room number I gave you last time, which was wrong, the correct room number is 2013, and the office hours will be right after the lecture, just to give me enough time to wash the chalk dust off my hand, and then we'll start the lecture. Last time we ended by talking about the algae. We said that the algae were a remarkable group of organisms which probably and surely were the organisms that innovated their life cycle by inventing the alternation of generation scheme. And to remind you about this scheme, we basically are going from a sporophyte, which is 2n, to a gametophyte, which is n. And this cycle continues with the alternation of these two generations of plants. Now, to give you some idea of the scale of time when this scheme was invented, I'm going to go back to our eukarya line that we had the very first day of lecture. Put on here humans at the end of the line. And now I'm going to insert the algae, and in particular we're going to spend most of our time talking about the green algae. And what we can say is that the alternation of generation scheme was developed during the first billion years of algal evolution. And it was probably invented a number of different times, not just once. So whatever was the selective pressure to develop this type of lifestyle, going from a sporophyte to a gametophyte, must have been very strong. Although we don't know what it is, several of you have asked me why we would have a second plant produced, why delay fertilization. There are lots of theories about why this came about, but no one really knows for sure why this lifestyle occurred. So during this first billion years, the algae were inventing and trying different types of sexual reproduction, and ended up inventing alternation of generations. Also during this first billion years, we think that an alga in the ocean, where life began, probably moved to lakes and bogs on the land. Now remember, the algae require water for fertilization. That is because of the flagellated gametes. And so if they were going to move to some place, they had to move to some place where they could continue their life cycle, where there was standing water, as you might find in a lake. And then after the first billion years, about 490 million years ago, we think a remarkable event happened. And at about 500 million years ago, or 400 million years ago, so we'll say 500 to 490 million years ago, an alga moved from a lake or the bog onto the land, became the first land plant. Now, imagine this organism here, which is a brown, but nevertheless is still an alga, moving from an ocean environment onto the land. What are some of the problems it would encounter in moving outside of a water environment? What do we have as sort of the problems? Yes? No physical support. Okay, so we have problems with support because it's buoyant in the water. What other problems would we have? Fertilization, okay, very good. Anything else? They dry out, very good. And there's one other big one which is related to drying out. Why they dry out? In the water, these organisms are surrounded, essentially, all the cells are surrounded by water and nutrients. So there is no reason to have to worry about transporting water inside the plant. So we don't have an internal transport system, no transport in the alga. And this would be a problem if you moved on to the land because only your roots would be in the soil. The rest of the plant would be removed from the soil where there would be a water supply. So what did the first land plants look like? What were those first plants like, and what were their characteristics with regard to moving on to the land? We think the first land plants which moved on to the land have characteristics of a group of organisms that we're going to talk about today, namely the bryophytes. The bryophytes are known to you as the mosses and the liverworts. And we believe that they retained many of the characteristics of their first algal ancestor, of the algal ancestor which moved on to the land. Now, what are some of the ways in which bryophytes have retained the characteristics or have dealt with the fact that the first algal ancestor moving on to the land had these problems? Well, the first characteristic is that the bryophytes have remained small. So they don't have to worry about transport. Now, even though they're small, they might be a couple of centimeters long, they still have to worry about water. And so for those of you who have collected bryophytes or will look at the slides or come up and look at the material here or in the lab, the way the bryophytes have dealt with getting water and nutrients and not having a transport system is they remain oppressed. They are tightly on the surface of the soil so that they can absorb water directly over a large surface area. The fact that they've remained small means they don't have to worry about support. Because they're small. How about fertilization? The bryophytes have retained from their algal ancestor flagellated sperm, flagellated gametes. This means that they have to have water at some part of their life cycle so that fertilization can occur. So we probably would find them in an environment, a periodically wet environment or moist. So these organisms that we're going to talk about very briefly today, the bryophytes, have the characteristics that we think were ones that were similar, or if not identical to, the first algal ancestor plant moving on to the land. Now, as I said, the bryophytes have to live in areas that are periodically moist. So let's think about this lifestyle. They have retained alternation of generations from the ferns, and we're going to talk about that in a moment. They require water for fertilization, and they have these other characteristics which are based upon their need to be able to survive on the land. Now let's first talk about the alternation of generation scheme in the bryophytes. In the bryophytes, unlike the algae, where we drew, and I'll write this for you in a moment, I want you to listen to this, where we drew the sporophyte and the gametophyte and we said nothing about their size, in the bryophytes, the gametophyte, or haploid generation, is the free-living photosynthetic generation. photosynthetic, largest plant in the life cycle. Now, how did this come about? When the algae were evolving during this first billion years, and we can even see this today in some of the algae in the oceans that exist today, sometimes the gametophyte was bigger than the sporophyte in some of the algae. Sometimes the sporophyte was bigger than the gametophyte. And sometimes they were both the same size. They were both free living in the ocean, and they could vary in their relationships. One would be bigger than the other, or maybe the same size, but they were both photosynthetic. But the first plants which moved on to the land, maybe they came from an alga in which the gametophyte was larger than the sporophyte. Those first land plants, the bryophytes, have the gametophyte bigger than the sporophyte. And this is an important point which we need to put in mind. And second thing is that the sporophyte, that is the 2N generation, is small and not free living. Small and not free living. I'll show you some pictures of this in a moment. So let's look at the bryophyte life cycle, and we're going to go from sporophyte. We'll start with our same place which we had before. Go through meiosis to the gametophyte. The point that I would like to make here again is the gametophyte makes gametes, which are N. And the gametes are eggs and sperm. Now let's think about this lifestyle. Here is an organism which grows in an environment that's periodically moist. Maybe it rains every year only for a month. Or maybe it rains only for a few days. This organism can't do any fertilization because there's no water to allow the flagellated sperm to move. But if this organism waited to produce the flagellated sperm until the water came, it's possible that they wouldn't produce it fast enough. In other words, they see the water, they start to make the gametes, and then by the time the gametes are ready and formed, the water has evaporated. So what the bryophytes have done is essentially they pre-make the gametes and wait for moisture. So what they're doing is preparing themselves to act immediately as soon as the moisture sort of comes on the market. But there's a problem. And that problem is that those gametes which are being made during a dry period of time risk drying out, risk being killed by the dryness. And so the invention of the bryophytes is to make protective structures surrounding the gametes. And these protective structures allow them to keep the gametes alive and not drying out until the moisture comes. And these protective structures are given two names. The one which protects the sperm is called the antheridium. It's like a jacket which surrounds, a layer of cells which surrounds the sperm. And the one which protects the egg is known as the archegonium. Now let's briefly look at the life cycle, and I'll make some drawings here, of a bryophyte. And let's say we're going to look at a moss, and you're going to see this moss in the lab. We're going to start off with the most conspicuous, obvious generation that we see in the moss. It has these little structures which are green, which look like leaves, And I'm going to make these structures. These are the gametophytes. The gametophytes. They are the green, free-living generation. These are the things that you see. If any of you come up afterward, or I'll show you slides, when you look at a moss growing on the ground, you generally see the spore of the gametophyte as being the dominant generation. So let's say this is the female gametophyte. So now we're going to have the sexes separated into two gametophytes. And this is the male. What does a gametophyte make? Gametes. Okay. So the male makes sperm, and it makes the sperm in an antheridium. And the female makes eggs, and she makes it, or it makes it, in an archegonium. And now, usually what happens is that there is a rain or rainwater which is flowing through, and this rainwater will splash onto the males and then may splash over again and carry the gametes. Or it's possible that the male gametes will swim on a coating of water and be attracted to the archegonium by, we believe, attractants that the archegonium produces. In any case, here is the female gametophyte again. And now we've had fertilization. So in this archegonium, we have the new 2N embryo, the new sporophyte. And if we allow the sporophyte to develop, I'm going to make the female a little bit smaller so I can put it in. Growing out of the archegonium, this is the female again. Growing out of the archegonium, depending upon the species of moss or liverwort it is, we find a structure which is the new sporophyte, 2N. Now notice in this particular example that I've given you here, it is not free living. It's like a parasite. It gets all of its nutrients from the female gametophyte. It's growing from it. It's never free living like the gametophytes of the algae. It grows from the gametophyte. So in this particular life cycle then, we have spores via meiosis, and the life cycle then repeats with the production. This is the life cycle of the bryophytes. An important life cycle. It has some contradictions because the gametophyte is bigger than the sporophyte, but the basic life cycle, alternation of generations, is the same as you saw in the algae, and it will be the same as we go up and look at the other groups of plants. Important life cycle, if you understand. You'll be spending time this week in lab looking at it. Now, the problem with this particular organism, as far as its being able to grow many places, is that it's really limited to places that are periodically moist and places that don't have other plants shading it out because they remain very tiny. So while the bryophytes are a successful group of organisms, they're rather limited in the different environments in which they can grow. The next evolutionary advance then, to allow plants to spread into other areas, involved the production of a transport system which allowed the plants to be able to grow in environments which were moist at the bottom, in which the plants could get water via roots or via some organs which absorb the water. So the next advance in the evolution of plants is the development of a transport system. And biologists, in sort of parallel to the human blood system, we call this transport system the vascular system. And the vascular system consists of specialized cell types which are involved in the transport of water and nutrients. We'll talk about those cell types more later on. But right now, I just want to introduce you to the notion that the next evolutionary advance involved the production of a way of transporting water great distances. This allowed the plants now to move into other environments, but as importantly, it allowed them to get big, because they didn't have to be close to the water supply as the bryophytes are. So I'd like to begin by talking about plants which are known as vascular plants. There are probably about 250,000 vascular plants, and they've spread to every environment on the Earth. And this is in large part due to the fact that they have this transport or vascular system. I'd like to begin to consider this large group by making an artificial, or not so artificial, but a division. And the division is going to be seedless plants and plants with seeds. We'll talk about what seeds are in a moment, but let's first begin our discussion of the seedless vascular plants. Now, probably the best example and the one that you are most familiar with of a seedless vascular plant is the ferns. And I have up here several different types of those 12,000 species of ferns. And what I want to point out to you is that the distinguishing feature of the fern is this large kind of leathery leaf, which is often called a frond. This is a common name for it. Now, this plant, and this is different from the bryophytes, this plant that I'm showing you in the front is the sporophyte. In the ferns, the sporophyte is the dominant generation. When I mean dominant, I mean biggest, or most conspicuous. Now, if this is the sporophyte, what do sporophytes make? Make spores. Where are the spores made on a fern, on this sporophyte generation? Well, many of you have actually seen these structures on the underside of leaves of ferns, and you'll have a chance to see them in the lab. On the underside of ferns are collections of dots, depending upon what species of fern we're looking at. And I'll show you some pictures of them in a moment. These collection of dots consist of many, many spores. So this particular leaf is a spore-bearing leaf. And we give it a technical name, and that technical name is based upon its function. We call it a sporo because it has spores involved with it. Fill, a sporophyll. So the fern that you see up here has these leathery leaves, which are fronds, and they are also sporophylls because they make spores. Now, are these spores just sitting here on the leaves, just waiting to be picked up by somebody or fall to the ground? In fact, on the sporophyll, the spores are inside of a structure. They're not directly exposed initially to the outside. So on the sporophyll, spores are inside a protective structure. And that protective structure is called the sporangium. So when you look at the underside of a fern leaf, you are looking at actually many, many sporangia, and inside each sporangium are many, many spores. So there are tens of thousands of spores on the underside of each of these sporophylls. This is the sporophyte generation. This is the dominant generation. It is the generation that most of you think about when you think about ferns, if you do. But ferns have retained the characteristic from both the algae and the bryophytes. That is, they show alternation of generations. What does the second plant look like, which is needed for this plant to complete its life cycle? In this vial or beaker up here in the front, I have the second generation. I have the gametophyte generation. The gametophytes of ferns, let's draw one for you. So we're going to show you the gametophyte of the fern. It's N, and it's often kind of heart-shaped, although not always. It's about, depending upon maybe 0.5 to maybe 2 centimeters in size, so it's tiny. It is photosynthetic, meaning that it is free-living, and it is the smaller of the generation. So for this fern, or for any fern to complete its life cycle, it has to produce this other plant, the gametophyte, to then be able to have the sporophyte produced. The cycle is only complete. Now, most of you will have never seen the gametophyte before, but if you look at some ferns growing outside, underneath those ferns, you might possibly see a gametophyte. So what I'd like to do now is to briefly draw the life cycle of a fern and include in that life cycle the sporophyte generation and the gametophyte generation. So now we'll look at the life cycle. And while this is going on, I'll just put the screen down since it takes such a long time. We'll begin, this is the fern now. We'll begin with the sporophyte. The sporophyte is the plant you see up here in front, and via meiosis on the sporophylls, the same leaves we looked at before, we make spores, which are now N. This should be exactly the same as you saw in the algae and in the bryophytes. And those spores now make a gametophyte. Which I said were these little teeny organisms. There are many of them in this particular jar here, and they're heart-shaped. Now let's look a little more closely at the gametophyte of this particular fern, whatever it happens to be. If we look carefully at this gametophyte, we'll find that it's very thin. It's green because it's photosynthetic, or it's photosynthetic because it's green. And it has kind of two thickened areas on the gametophyte, and you'll be able to see this in the lab. In one of these areas, we have archegonia with the eggs, the haploid female gametophyte, female gamete. And the other one, we have antheridia, and there usually are many, which contain the sperm. Now, these sperm are flagellated. They have flagella. So this is a characteristic that they have retained from the algae and from the bryophytes. So they need water. They need water to affect fertilization. So let's see what would happen in fertilization. We could have a sperm, once water is available, swimming to the egg and fertilizing it. But if we did that, we wouldn't have any genetic recombination because we're essentially fertilizing ourselves with the same thing, same DNA that's in the egg. So what many gametophytes do is they first mature the egg and wait for sperm to come from someplace else to allow for genetic variation to be introduced into the plants. Once they're fertilized, they then release the sperm, and they then have that sperm swim to another gametophyte to affect fertilization. If by chance this gametophyte is by itself and there's nobody else around, rather than just dying out and losing the genetic material, it'll fertilize itself. But in that case, it produces an individual with no genetic difference from the parent, but the organism is able to continue its lifestyle. Now, let's take a look at one of these gametophytes after fertilization has occurred. And I'll show you a slide of this as well. So we've had fertilization. Fertilization has occurred. And so we're going to look now at the gametophyte, and I'm going to draw for you just one archegonium. One archegonium, although there are usually many. In this archegonium is the new 2N embryo. New 2N embryo. This new 2N embryo is going to develop into a sporophyte, a new plant, new generation, just like you saw before. And what you find happening is that as the embryo develops, becomes more organized, that a new fern grows out of the archegonium. A new fern grows out of the archegonium. And this then results in essentially the death of the gametophyte. It provides nutrients for a very short time to the developing sporophyte, and then the gametophyte dies. Its function has already been served. It is allowed for the gametes to be produced, and it has sort of supported the new sporophyte. And so I'll show you a picture very shortly of a new fern which is growing out of the particular gametophyte. Now, what I want to point out to you is that if you look at the size of this gametophyte, and being very tiny, as I said, half a centimeter to a couple centimeters, it's also very thin. As the gametophyte has gotten smaller, much smaller than the dominant sporophyte, but yet free-living, it's encountered a problem. And that problem is that there are less cells and tissues available to make up the archegonium and the antheridium. So as a result, we now are impacting on the safety of the gametes. And after I show you the slides, we'll go back to this point about what the next evolutionary advance does to make up for the loss of the tissue, which has now been reduced in the archegonium and the antheridium as a result of the gametophyte getting smaller. So let's look now at some slides here of both the bryophytes and the ferns. So this first slide is of a bryophyte similar to the one I've drawn on the board here. These structures here are, as you will see, here is the gametophyte part of the plant, the photosynthetic free-living part, and here are the sporophytes growing up and attached to the gametophyte, and spores would be produced in the tops there. Here's a liverwort, probably a less familiar plant to many of you, and what I want you to notice here is that we're looking at the gametophyte. It is tightly oppressed to the soil, so it doesn't need roots and it doesn't need a transport system. It can absorb the water in the soil by being in direct contact with the soil. Here's another example of the slowly growing but also slightly oppressed to the soil gametophytes. Here are the gametophytes growing against the soil, and you're going to see these umbrella-like structures in the lab. They are involved in sexual reproduction. The majority of the green tissue that you see here, and in fact even these umbrellas here, are all gametophyte tissue. Only under the arms of the female, after fertilization has occurred, do we find the sporophyte, which is a parasitic, tiny 2N plant which is attached to the gametophyte and which grows. This is a very important moss. This is sphagnum moss. It is used worldwide. It's estimated that 1% of the Earth's land is covered with the peat bogs with various mosses. Now, let's spend a few minutes looking at the ferns. In the ferns, in contrast to the bryophytes, the dominant generation is the sporophyte generation. These are sporophylls, and under the undersides of these sporophylls, meiosis occurs. Spores are falling to the ground and making the gametophyte generation. And if we look at the underside of one of those sporophylls, we see these collections of dots. What you're actually looking at are sporangia. So if we look at it a little higher power, each of those dots represents a sporangium, but there are many sporangia here. And inside each of these sporangia, which is a little higher power, we have many, many spores, many, many spores. If the spores are allowed to germinate Here are the gametophytes, these heart-shaped structures. These are the structures on which the sex organs form. And just to give you an idea of their size, here's a test tube, and here are 3 or 4 gametophytes. They're photosynthetic, they're free-living, but they're very tiny. When water is provided, the male sex organs, that is the antheridia, can release the sperm, and these are flagellated sperm which are being released in the water, and they then swim over to the female portion of the gametophyte, and in this particular slide here, we have an archegonium. Here is the egg, and here is a male gamete sperm just before fertilization. This occurs on the gametophyte, so that finally what you see is a gametophyte, and if we could have traced this back, there would have been an archegonium here, and from this archegonium, after fertilization occurs, this is the new sporophyte, the new plant beginning to grow, just as we have up here in the front. So these are the ferns and the bryophytes. The important points that you guys should keep in mind about them is that they still both need water for fertilization. They both have alternation of generations. These are a table that I would suggest you make. These are just a review. And in the ferns, the sporophyte is dominant. Its gametophyte is tiny but free-living. In the bryophytes, the gametophyte is dominant. The sporophyte is not free-living. So you have the same cast of characters, and what I'd like you to do to help you study this is make up a table with these various characteristics. And you'll see there's very little memorization involved. It's an understanding of how things changes in evolution. Now, I said before I showed you the slides of the ferns that one of the problems is the gametophyte got smaller and smaller, and as a result of getting smaller, there was less tissue in the archegonium and the antheridia to protect the gametes. The next evolutionary step appears to have been pushed by the fact that the development of extra protective structures for the gametes allowed the next group of plants to become more prosperous and to spread into environments and in climates where the ferns could not grow. And that next evolutionary step was the development of a seed. So we're going to talk now about vascular plants which make seeds. And underlying this discussion, where we would like to end up today is, where does the seed come from? Plants with seeds, or seeds, we think evolved probably about, so many people think about 360 million years ago, the first seed-bearing plants evolved. So they coexisted with the ferns, with the plants that were vascular, but that didn't make seeds. Now, they probably coexisted, we think, for maybe upwards of almost 100 million years. But about 250 to 280 million years ago, the Earth became a very hostile place to live. There was lots of glaciation which occurred, and lots of drought, dry weather which occurred. Therefore, organisms which had an advantage in being able to overwinter or to protect their gametes during these stressful periods were able to prosper at the expense of those organisms which were confined to smaller, more temperate areas. And it was during this time that the first group of plants that we want to talk about, first group of plants with seeds, began to dominate the environment. And that first group of plants that I'd like to talk about, first group of seed-bearing plants, is familiar to you, and it is a plant that you probably see every day. Those first seed-bearing plants, the group that we want to talk about now, are plants which are known as the conifers, or you may know them as the cone-bearing plants. Or the technical term for these plants is the gymnosperms. These are seed-bearing plants. So plants that would be familiar to you as redwood, the pines, plants which produce these structures that you know of as a cone. These cone-bearing plants are the ones which make up the conifers or the gymnosperms. Now, what does the word gymnosperm mean? Why do we apply this term to something that has the word cone-bearer on it already? It literally means naked seed. I'd like you to all put down your pencils for a second and just look at this demonstration here. Here is a cone of probably a Monterey pine tree, a cone-bearing plant. If I take this cone and I turn it upside down, the seeds fall out. And they fall out because the seeds essentially are not surrounded by any other protective structure. They're naked seeds. So this is where the term gymnosperm comes from, a naked seeded plant. Now, the gymnosperms are an important group of plants. They have spread to many, many different environments. And what I'd like to do is now to spend some time talking to you about the gymnosperms and talking to you about their life cycle. So, first of all, let's begin with the fact that the gymnosperm tree that you see outside that produces a cone is the sporophyte generation. So we can at least say from our start of our discussion, we have the sporophyte generation. Now, this structure is a cone, and you know that the cone is produced on the plant growing outside, which I've told you is a sporophyte. Now, going back to our theme of trying to understand where the seed came from, I want to return for a moment to this fern and compare it to this cone. If this is a sporophyte, and it's part of the sporophyte, what does the sporophyte make? Spores. Okay, where are the spores made on a gymnosperm? In order for you to understand this, I want to take you back to this fern, and I want you to imagine that we take all of these sporophylls, these spore-bearing leaves that are part of this fern, and we collapse them down into a structure which is this cone. So each of these scales on the cone would represent, in this case, one sporophyll. So a cone is really a collection of sporophylls. And if you want to look at how the cone originated, through evolution, a large number of sporophylls collected and made up this cone. So if these are sporophylls, what would be produced on each of these scales? Spores. So this is where the spores are produced in the sporophyte generation of the cone-bearing plants, the gymnosperms, on these scales, representing a collapse, so to speak, of these particular sporophylls on the fern. Now, this is the cone that you think of when you see a gymnosperm, this large woody cone. But in fact, if you look at a gymnosperm carefully, especially during the time when reproduction is occurring, you'll actually see that there are two cones, two different kinds of cones. There is a large cone, such as this one, a large cone. So we have two different types of cones. There's the large cone. And the large cone makes spores. And because they're made on this large cone, and because of other reasons, we call these spores megaspores. And we'll talk more about them in a moment. And then there's a small cone, which most of you never see or never realize, although I guarantee many of you have experienced the product of this small cone. The small cone is very transitory, and the small cone also makes spores. It's essentially the same type of structure here, but it's a small cone. It makes spores, and these spores are called microspores. But you better know the microspores as pollen. So the pollen has been released from these, and how does the pollen get from here to the other cone where fertilization? How does the male get to the female? By the wind. So here we now have an example for the very first time in which the transfer of the product of the spore, namely the gamete, doesn't depend on water. It depends on the wind. So in this particular example, you see a big evolutionary advance. We no longer need freestanding water at some part of the life cycle for the transfer of the pollen. But because we're depending upon the wind, and because the wind is such an unreliable messenger, we have to produce many, many millions and billions of spores to affect fertilization. And so it's estimated that for each successful fertilization which occurs, you have to produce 1 million spores for plants that depend upon the wind to carry the spores from one place to another. Now let's look at the life cycle of the gymnosperm, putting onto it a couple of these characteristics that we've spoken about. What I'd like to do is to draw the two different cones for you so that you can see what's going on in each of them. And the first one we'll do is we'll begin with the male cone, the smaller of the two cones. This is on the sporophyte, and it consists of scales. So if we looked at one of these scales, this is essentially a sporophyll. It's a sporophyll because it is a spore-bearing scale. And so we have spores being produced. So we have to have meiosis occurring here. Meiosis, the same thing you saw in the ferns. These spores then, after having undergone several divisions, will develop into a pollen grain or a microspore. You have a chance to look at them in the lab, and often in the gymnosperms, the microspores have these two, looks like Mickey Mouse ears on them. These are to help it carry in the wind, and it may consist of a couple of cells depending upon the species. So this is the pollen grain or microspore. Now let's look at the female cone. The female cone is the larger of the two, having these scales. Let's take one of these sporophylls, and on these sporophylls then, we now have a structure which houses the spores. What is that structure that houses the spores called? We just saw it in the sporangium, right? Sporangium. We call it a megasporangium because it is for the larger organism. What does a sporangium make? Just told me. Spores. So inside here we have a spore. Okay. Here's that scale again. What does a spore become? What does a spore germinate into? What does a haploid spore become? Which plant? Gametophyte. Okay. So here we have the megasporangium with a spore that has germinated into a N-megametophyte. Now let's take a look at what's going on that scale. Make it big for you. Here's that scale again. Here is the megasporangium wall. Now let's draw a picture of the female gametophyte. The female gametophyte. Just like you saw before. It has in it a archegonium and an egg. Same thing that you saw before. Archegonium and an egg, nothing different. And now we get fertilization. Now the pollen grain comes over. The male gamete is released, and we get fertilization. So let's see what we have here. Let's draw our scale again. This is the scale. On the scale now, we have the megasporangium. This is important because we're leading to where the seed comes from. We have the new sporophyte, which is 2N. And now this is the secret of the seed. Surrounding the megasporangium wall, a new structure develops. People aren't quite sure where it came from. This new structure is a seed coat. This new structure, which developed to protect the developing embryo, to protect it because in the ferns we had less and less tissue available. And so we can define a seed as a megasporangium. This is at its most basic. Megasporangium surrounded by a seed coat. So the seed developed as a result of this new protective structure to keep the embryo safe during bad times. Okay, look at these life cycles, make your table, and it'll be clearer to you, and you'll see you will not have to memorize very much.